Hi, everyone. Welcome to the Live Longer World podcast. Through this podcast, I have conversations with scientists, biotech entrepreneurs, investors, and other advocates that are transforming the field of longevity science. We have conversations on how you can slow aging, reverse aging, and maximize longevity now and in the future. If you are new here, my name is Astha Jain and I am an advocate for longevity science. Besides this podcast, I also have a newsletter where I often discuss longevity lifestyle hacks and some basics of longevity science. On my YouTube, I also create some other videos talking about aging as well as longevity lifestyle tips. If you wish to listen to this podcast on other platforms, you can find it on Apple Podcasts, Spotify, or whichever other medium you prefer. All right, that's it for now. And with that, let's get started with today's episode. My guest today is Dr. John Eber, who is a professor of neuroscience and genetics at the Albert Einstein College of Medicine in New York. Dr. Eber was fascinated from a young age by how a single cell forms the entire human body following an innate genetic program. And he got excited by how knowledge of this process might be used to improve our biological fates and defeat aging. He is a molecular geneticist by training and obtained his Ph.D. from the University of California, San Francisco, before specializing in the study of how neural stem cells form the brain at Stanford University. His lab is now developing ways to use cell and tissue replacements to repair and rejuvenate the brain. More specifically, the Eber Lab is engineering tissue that increasingly resembles normal neocortex, which is the part of our brain that forms our highest cognitive functions, and testing the ability of this tissue to integrate with the host brain. Dr. Eber is also the author of the book Replacing Aging. In this conversation, we had an extremely interesting discussion ranging from why Dr. Eber thinks the other methods to fight aging may not work if the goal is to beat aging. We also discuss 3D organs and tissue cell replacement. And we talk about the neocortex and what it takes to replace one. And lastly, we also discuss how plasticity in the brain works. Even if you're not associated with the field of longevity, I think you'll find this conversation interesting and very refreshing. Radical to say the least. All right, with that, here is the conversation for you. Professor Eber, thank you so much for joining me today and welcome to the Live Longer World podcast. It's such a pleasure to have you here.
Thank you for having me. It's a great honor to be on your podcast. I appreciate the opportunity.
Absolutely. Well, the reason I'm excited to talk to you is because you have a very radical way of approaching how we can fix aging. And just to set the foundation for the listeners, your approach is around replacement of each of the components of the body, including the brain, which we will go into, as a way of entirely curing aging. And of course, I'll let you chat more about it. But I think a good place to start would be how you actually came to this conclusion that this might be perhaps the most effective way to cure aging. And I know, especially in your book, Replacing Aging, which everyone should read, you talk about why some of the other approaches, such as, for example, just clearing away senescent cells or fixing mitochondrial dysfunction, they are great, but they're not going to entirely cure aging. So it would be great if you could talk about why some of those approaches, in your opinion, are at best just going to maybe slow aging.
Yeah, great. I love the way you asked the question, that it's a radical approach. And I have to say, I'm somewhat conservative by nature. And so to me, I see how it comes across as a radical approach because it involves surgery as opposed to just taking a pill. But it's not so radical when you consider all the scientific evidence that we have for what aging is. And that's where I think the other approaches, although maybe very useful in making us healthy or fighting certain age-related diseases, will be much less effective, or I would argue not effective against reversing aging itself. And the reason I say that is because it's been pretty well documented, all the different forms of damage that occurs to the molecules that make up our body. So those are proteins, DNA, carbohydrates, and fats. And they all accumulate damage with time. And these forms of damage are known, at least for the DNA and protein damage, are known to cause the other hallmarks of aging that people are focused on, which I think are downstream of the real problem, which is this accumulation of damage. And the tricky part is that this damage occurs not just to the cells that make up our body, but to the environment that the cells are in. And that's perhaps the most neglected area of longevity research, because much of the focus is on the cells and not the extracellular environment that they live in. And our bodies are largely made of that extracellular environment. Yes, all our tissues have cells, but all those cells are nested in this extracellular space, sort of connective tissue, but it's very functional tissue and it's very different from organ to organ. And it accumulates a lot of damage over time that is not addressed by metabolic approaches, because those remain in the cell, not addressed by, pretty much all the approaches that are very popular right now. Even epigenetics, epigenetics is great, cell reprogramming is great. It has a lot of uses. It can be used for regenerative purposes, but it can't be used for reversing age-related damage, because cells respond to their environment. And if they're already in an environment with all the extracellular matrix they have, they will not produce more. And even if they did, they would just be adding more protein to the damage that's there, and that wouldn't be very functional. So this extracellular damage is particularly problematic with the approaches that are currently getting much of the attention. And the cellular damage itself is also somewhat problematic. I mean, there are some longevity researchers who would argue that within the cell, the DNA damage itself is in many cases irreversible because of the loss of information content due to mutations in the DNA, and that accumulation of damage over time is also a big driver of aging. I think it's one of the drivers of aging. I think all forms of damage to all our classes of these macromolecules, the proteins, DNA, fats, and carbohydrates, are all going to be on their own sufficient to bring us to a decline with time and inevitable demise, but unless, of course, we reverse them. So these forms of damage that occurs to these classes of macromolecules are stochastic and complex. And so imagining ways of reversing each one of them with an enzyme or a drug without causing worse side effects than contributing benefits is very difficult, because there's hundreds of forms of damage, and even the best drugs we've developed today have side effects. So I can't imagine taking huge combinations of drugs and thinking we're going to be better off than we were before. So really, I've come to the conclusion, and I guess we'll talk about this more as well, is to really beat aging, we have to replace tissues or organs or body parts, but not just cells, not just target specific pathways, but replace entire tissues.
I see.
Yeah.
Okay. No, that was actually very helpful in explaining. So I heard two main points. One, you're saying is that a lot of these approaches to even reversing the damage is mostly restricted to reversing damage within cells. And there is this extracellular matrix around, which is the environment around the cells that accumulates all this damage. And it sounds like not a lot of people are focused on that. And that is also something that we have to fix. And then I think point number two was that even if we have all these combinations of approaches, which was actually going to be one of my questions, that, what if I try to fix damage in all the different tissues? Then, I mean, in theory, maybe I take like 50 pills, sure. But if I could fix all the damage in all the different parts, then why could that not work? And you're saying that maybe it could work, but it's just going to be extremely complex because obviously biology is interrelated. You don't know what effect one pill would have on something else. How do you fix the damage in each of the different regions?
Exactly. It's that complexity that I think, and maybe I was too optimistic. I really don't think it can work. Again, the reason is the complexity of the damage, but the drugs, or some of these are covalent modifications. So some drugs can act to break covalent bonds, but again, those are usually very toxic drugs because they cannot recognize the difference between a covalent bond that serves a purpose in a protein versus one that happened stochastically due to damage in a protein or any other molecule. So these reactive drugs tend to be very toxic. And so now you would want ones that are smart enough, and drugs are not smart. They can't compute anything. They're small molecules, typically. You would want them to just target the bad covalent bonds and not the good ones. And that's just not imaginable. Enzymes are a little bit better at specificity, so they're much bigger molecules, and you can have them target specific forms of damage. And there are groups that are trying to do this, and there may be benefits that they're able to obtain. But again, they're going to be limited because even enzymes are not that smart. Even if they can recognize like a form of glycation to a certain protein that's bad, they might be able to recognize to some extent that it's bad. But then, glycation occurs to proteins under all different contexts, all different proteins. So it's going to look different in many cases. And there's forms of glycation that are good and that cells use for important purposes. And those have to be left alone and not touched. So even the enzymes, it's going to be very difficult to come up with a battery of enzymes that are effective that way. So I'm rather pessimistic about these approaches as far as really reversing aging and beating aging, so that you can make an old person really like a 20-year-old, or again. But again, that's not to say that there won't be some benefits from drugs and other forms of biological therapeutics like enzymes, or there are other forms as well. So I think they're worth pursuing, but we need to recognize their limitations. And if our goal is really to beat aging and reverse aging, then again, I think the most straightforward way to go about this is tissue replacements.
Right, and I think that's a very important distinction to make, that, as you say, if we really want to beat aging, then your approach might be one of the best ways to go about it, maybe as of now. But because, I mean, some of the other approaches, they could be effective. Maybe we end up living to be 150, 200, at least not having diseases when we're older, but then we're just essentially pushing the timeline as to when those diseases will eventually catch up. And how long can you keep pushing that timeline is I think where your argument holds.
Yeah, 150, 200 is pretty optimistic. I mean, we all know 90-year-olds and very few 100-year-olds, and we know what they look like and how fragile they are. So, and they could be disease-free for their age. They'll be disease-free. So, and the decline is not necessarily linear at that point. It may be a little accelerated. So, I think 150 is, unrealistically optimistic.
In a few years, I think we'll get there.
With just treating diseases, with just eliminating.
Right. So, yeah. All right. So I think let's dive into what research you are doing around tissue replacement, and then also discussing the brain, because I think that's probably the more complex part. But we can just start with the basics on how you're approaching beating aging.
Yeah. So. I wanted to sort of, because it's a neglected area in the field of aging, but it's not neglected in regenerative medicine. I mean, people are fabricating body parts, growing organs in the lab. So this is a very active area of research and development, but there's very little overlap with the longevity field. And I thought, this is ridiculous. This is really what I think is going to work for aging. And so we need to be talking about it. So I wrote a book about it. And the funnest part of writing the book was finding out just how much has been done in body part replacements in humans. I didn't realize. But essentially, every part of the human body except the brain, and we'll get to that later, has been replaced in humans surgically. So the implementation is possible. So, and when I say every part of the body, it really is every part of the body, like face, scalp, nose, hands, limbs, flank, ribs. I mean, every internal organ, sexual organs. Everything's been transplanted successfully in humans, but in most cases, they've relied on transplants from deceased individuals, right? Organ donors, which is, and those are very, very precious, because those transplants are used to treat people with fatal critical diseases to save their lives. So that's what they should be used for. So they can't be used for just reversing aging. And in some cases, it's due to terrible disfigurements or loss of body parts. Transplants will be used in those cases as well. Again, there's a greater need for those individuals than there is for somebody who wants to be 20 again. Mm-hmm. Not that there's anything wrong with that. I'm all for that, of course. But we need to prioritize. So what we need is the development of lab-grown organs and or prosthetic body part replacements. And both of those developments are being made that are pretty astounding. There have been some organs that have been grown in the lab that have been transplanted in humans and have been functioning for over a decade. The bladder is one example, but it's not the only one. There are others as well. And so that's very promising. And every other internal organ, for sure, is in preclinical development. So I think it's inevitable that soon enough, we'll be able to replace our essential organs with transplantable ones. There are different ways of growing in the lab. There are different approaches being used. For example, some people are making pigs that can grow human organs, for example. So there's all sorts of approaches that people are using. And there's no reason why they won't succeed. They just have to work out the immune compatibility in that case. for the lab-grown ones, one of the biggest challenges is the vascularization to scale those organs to get bigger, but progress is being made there too. So I think it's inevitable. So those are biological replacements, but there's also prosthetic replacements. We know the heart is an obvious example. People have been living with artificial hearts sometimes for over a decade. So, you know, there's already that, you know, that's already a good example. There was just recently at UCSF, University of California, San Francisco, a group that developed an artificial kidney. And you know, kidneys are in great demand for diseases, but you know, can also be used for rejuvenation purposes, especially if they're, you know, artificial. They can be mass produced, and so you know, that's another example. And there are, of course, for limbs, prosthetic limbs. There's, you know, prosthetic hearing devices. There's prosthetic vision devices, or artificial eyes or retinas that have shown functionality. So all these things eventually will lead to the ability in the not too distant future to replace all body parts functionally, so that we can, you know, when we're getting old, we don't have to suffer because our heart is weak or our kidneys are failing. We can get high-performing young ones again. Same with our limbs and basically every body part.
That's quite exciting, that as you mentioned, every body part can be replaced. I mean, obviously from a regenerative medicine perspective right now, but also from like, it's nice to know if I start getting old and one of my organs fail, that can probably just swap it out. And of course, it's one of the reasons is because it's possible to now make some of these artificial lab-grown organs as well. So I think that could definitely accelerate the field. All right, so where does the complication lie then? It sounds like the brain.
Yes.
Yes, of course. So what is, because you focus on the neocortex, but I guess just more of a high level overview, how would replacement in the brain work? And maybe actually before that, a question I had was, do we know what aging in the brain looks like really? And is it very distinct from aging in the body? Like, would you categorize aging in the brain also as damage accumulation, or is there something else fundamentally going on?
Yes, it's definitely damage accumulation, and it starts occurring very early in life. The reason that there's sort of this misconception that, you know, we can have highly functioning brains even though our bodies are very old and fragile is because of the way the brain works. There's a certain level of plasticity that's inherent to how the brain works. And so that plasticity will compensate to a large extent for this accumulation of damage and for the loss of complexity of the neuronal connections that occur. So if you look at old brain cells, the neurons in the brain, they look very different than young brain cells. They're much less complex in their projections and connections and informing the circuits. But the nature of how they work is still very plastic. So that is what sort of masks, in some people's minds, the aging of the brain. But if you look anatomically or even physically at the brain over time, so in mice, it's very easy to do this because they have a shorter lifespan. You can look at their brains over the course of 2 years. Already by 4 months, which is considered young adult still in a mouse, there is an obvious physical difference in the ability of light to penetrate the tissue. If you remove the skull and you use like 2-photon imaging, which is this, you know, very fancy way of looking deep into the brain, already by 4 months, it's harder to do that than at 2 months. And that's because of the accumulation of protein aggregates and damage. So, aging happens in the brain, I think, just as fast as anywhere else, except it's maybe not as noticeable until very late when, plasticity can no longer compensate for the deterioration of the circuits that are there.
So when you say, plasticity, you mean that the functioning areas of the brain are compensating for the damaged ones, or the non-functioning ones? Is that why we don't really recognize aging in the brain?
Yeah, I mean, the damage is sort of widespread. It occurs sort of everywhere in the brain. And so what plasticity allows in older individuals, and you can see this by doing functional MRIs of older individuals, is they use broader and broader areas of their neocortex. That's the part of the brain that we use to think. It defines who we are as individuals, our self-identity. They use broader and broader areas of the neocortex for what they need, for their routine tasks every day. That may include, language and other motor skills and functions. So they may appear, to be functioning normally, and they are within their now more restricted behavioral, confines. So there's a lot less they can do, but they may still be able to do it just as well as a young person. They're just using broader areas of the neocortex. If you look at a young adult brain, everything is very compartmentalized, used very efficiently for different functions. In the elderly brain, that's not the case. It's more diffuse, more widespread, again, because the substrate has deteriorated. The neural substrate has deteriorated. So plasticity is just doing what it needs to do. If the person, uses a certain function every day, that's what plasticity will expand and encode in the rest of the brain. So that's this concept of plasticity and use-dependent allocation of neural substrate is really important in thinking about tissue replacement to rejuvenate the brain as well. And we can talk more about that as well.
Yeah, no, I absolutely want to dive into that. So I guess what you're saying is that over time, progressively, that even though, an older person, they might be functioning the same way, their brains might be functioning the same way, it's just less efficient. And then over time, as you age, even that efficiency in some ways declines, or the bandwidth of your brain basically dwindles. And which is why you start to notice loss of memory or loss of different functions in the brain.
Yeah, the efficiency in terms of encoding a wider range of functions declines, yeah. Efficiency for any particular function can appear or can remain the same as a young person, because now, more of the brain is devoted to that function. So you don't really see a difference compared to a young person.
I see.
But overall, yeah, you're less efficient.
Okay, that is very interesting. Before we dive into tissue replacement, you mentioned aging in the brain starts very early. What is very early in humans?
So, there's obviously less, we have less of an ability to look inside the human brain. Like I gave an example earlier with, looking at 2-photon microscopy. As far as I know, that's never been used on human brains. So we would be left with post-mortem tissue that can be analyzed. And I'm actually, I'm not that familiar with these careful analyses of brain tissue over time. So previously, biochemists have looked at damage. Again, I don't know how often they've looked in humans compared to other shorter-lived animals, but they've looked at damage over time to all different organs, and seen that there is this accumulation of damage. But for the human brain, yeah, I don't know, but certainly some forms of damage start accumulating maybe even before you're born. So because some proteins, like elastin is, the canonical example, doesn't turn over for a whole lifetime. So when we're growing as an embryo and a fetus and a child, though the elastin that's produced stays there for life. And that elastin, as soon as it's made, linearly starts to accumulate damage over time. Certain forms of damage like racemization is very linear. It's a physical change in the amino acid conformation, over time, and it's independent of biology. It's just a physical form of degradation, and that starts occurring right away. So then there are many other forms of damage that occur to elastin, breaks, over time, aggregation, glycation, different forms of oxidation. And elastin is an important example because it's in every part of our body, including our brain. So we're not just, the brain is not excluded from accumulating damage from a very young age. Elastin is in all our blood vessel walls, and it takes its name from being elastic, at least when it's young, but it loses that elasticity as we get old. So our blood vessels lose their elasticity and are no longer able to meet the changing demands of tissues. So I don't know if you know how functional MRIs work, but it's basically looking at the function of your brain in live, as it happens. And one of the ways it does that is measuring the flow of glucose or blood, and you can detect differences based on how much circulation is going on in different parts of your brain as you're thinking about different things. And so that's due to the plasticity of the blood vessels. So they meet, blood vessels are always dilating and restricting based upon the needs of the tissue. And with age, we can't do that anymore. And so that causes all sorts of secondary functional deficits in those tissues, including the brain again. So we can be pretty confident that, the molecular damage that accumulates throughout the body also, in some forms, accumulates in the brain from a very young age.
Oh dear, that's certainly not what I wanted to hear. So I was curious. I was like, well, am I already accumulating damage? It sounds like yes.
Yeah, but thank goodness for plasticity and our ability to compensate. So what's important to you and you keep using and thinking about, you can actually improve upon even when you're old because of plasticity. So not all is lost until the end.
That is good to know. And also hopefully, well, your work saves us, right? So let's talk about tissue replacement in the brain then.
Yeah. So tissue replacement obviously has to be done progressively. You can't, like other organs of the body, you can replace wholesale, and it's fine. You can replace your heart, your lungs, your kidneys, anything, and it's still you. Your brain, you can't do that. Your brain is you, and so the only way to replace it is progressively over time. But because of this plasticity, and that functions, if you're using them, and they're important to you, functions and memories will move from one part of the neocortex or one part of the brain to another over time. So because of that, the nature of how the brain works, this plastic nature of how the brain works, that means that we could replace tissue progressively over time without losing who we are as individuals, without losing our self-identity, without losing our memories, or our ability to perform any tasks that we like to perform. So that should be possible in principle. And it should also be possible in practice, because we know how the brain normally develops from precursor cells, so we have a pretty good understanding. Um, and at least we understand that the precursor cells know what to do and how they do it, even though we don't have, the finest detail understanding of all the molecules and genetic pathways that control that. But we know that they can do it, and we know that these cells, how these early brain precursor cells form brain tissue. And that's essentially what my lab and others are working on, is how to get these very immature brain precursor cells to form entire new tissue. And so the difficulty comes from the fact that there are many brain cell types that we need to generate to form a functional tissue. There are broad classes of excitatory neurons and inhibitory neurons. And then there are multiple support cell types, different types of glia, which are referred to as support cell types. Although my friends who work with glia don't like to think of them as support cell types, because they do more than that. They actually participate in, in circuit function, neurotransmission of electrical processes. So they're actually very important for that. So it's sort of wrong to just refer to them as support cells, but for simplicity, I'm going to refer to them as support cells. So there's different classes of those. And then, of course, there's also the vasculature that, as I mentioned, is obviously very important for any tissue of the body for it to remain young. So all those cell types form a tissue. And the trick for us, even though we know all the precursor cells that give rise to those final cell types, what we have to work out is the ratios of those precursors that we have to mix together, and their relative maturity states, because those cells are very immature precursors that are always dividing initially, and then they differentiate and give rise to neurons or these other cell types, and they no longer divide. And so we have to figure out, what initial ratio of precursors we need to mix to get the final ratio of cell types that makes a truly functional brain tissue. And we also have to organize them spatially. So these cell types are not just, all mixed together, but in the neocortex, for example, they're arranged, at least the principal neurons are arranged in layers. And so we have to have our tissue develop in such a way that those layers form as much as possible, mimicking how they form normally when we're fetuses and newborn, early in life, so that they wire correctly. So that's our challenge, to get that to work. But there's no reason we can't. I mean, we've already had early success in my lab, and others have also shown that the neural precursors do remarkably well in the adult environment. So they'll project very long distances to normal targets and wire up correctly. So this is hugely encouraging. And what we've done as well is put in young vascular cells and show that they'll form vessels that fuse with the host vasculature and circulate blood through the tissue that we're making. So this is all encouraging, but we're not there yet. But I think, again, it's a matter of time before we come up with the right ratios of cells and arrangement of cells to get a tissue that is fully functional, and at which point we can introduce it into the aging brain. And then there's a whole other side to replacement. So it's not just putting in young tissue, but we have to remove the old tissue as well. And that will probably be easier because we already know nature's already provided us with examples of how that happens by just progressively killing tissue, for example, in the language center. We know what rate the tissue needs to be killed at in order for language to be preserved by moving somewhere else. But we, of course, wouldn't do it the way nature has shown us, because the examples come from disease cases like the slow growth of a benign tumor. We can do it experimentally, even with light-induced silencing of neurons. So very precisely over time control the rate of silencing of brain tissue, allowing the function to be preserved by moving to a new substrate. So that at that point, that tissue is no longer useful to the individual because it's silenced. It no longer houses any functions or memories, so it can be removed. So that's the second part of the equation. So we have to make the tissue, which I think is the harder part, and then the silencing and removing of the old tissue is the second part. And the two hand in hand, done progressively, I think over the course of, we estimate 20 to 30 years is enough time for plasticity to move all the functions from old tissue to young tissue. We could have the brain of a 20 to 30-year-old again, and not worry about it for another 50 years. So that's what we plan on doing to really beat aging, including for the brain.
Right. No, that's actually extremely interesting. So you're saying we first use these precursor cells to create new tissue for the brain, and then use these, and then replace the old tissue with the new tissue, and all done in a progressive way. My question is, in the second part, when you say you can come up with these precursor cells and make the new tissue, which I know you said is the harder part, but say in a few years we get there. Once you start replacing the old tissue with the new tissue, especially because it's going to be done in a progressive way, how will the interaction between the two work?
Well, so again, this is all based on plasticity. So as the old tissue is being silenced, and as long as you're using those functions or recalling those memories, they will move to a new part of the brain, right? So
sorry, but that you would only, in theory, be able to recall them if there is still some functional element of the old tissue, or are you saying that a tissue that's still sort of working? I mean, it's obviously not in the best condition, let's call it, but then it has some memories attached to it, and then you can just move those to the new tissues once they are in your brain.
Yeah, so we have billions and billions of neurons in the brain, and memories and functions are encoded by minimally tens of thousands, but probably hundreds of thousands of neurons in each case. And those memories and functions are relatively fault tolerant, meaning you could remove a few neurons or add a few neurons, and you can still recall the memory. You can still use that function. And so what you're doing with this progressive silencing of a brain area, and you do like one area at a time where you know what the functions are, as long as that silencing is progressive and you're using those functions or recalling those memories, they will get re-encoded more robustly elsewhere without you losing them. So, there's this really nice example I alluded to earlier with language. So, language is something very important to us, and very, very complex. And yet, we know from examples in the literature that even though the original language centers progressively destroyed over the course of around 5 years, from a slow-growing benign glioma, the individuals never lost the ability to speak because, they speak every day, and that function therefore moved to a new part of the neocortex without anybody noticing any difference in their use of words or their ability to speak. And so, that we plan on using the same approach of progressive silencing. So, that brain tumor started off as just, one cell gone rogue and starting to divide, and eventually ate away at the whole language center to the point where after they remove the tumor, there's a big hole there, but the individuals can still speak. So plasticity is, the basis for why this works, as well as, memories are not encoded by single cells or neurons. They're probably fairly large networks.
I see. Okay, that makes a lot more sense. That is very interesting. And then I guess once these new neurons or tissues are inserted, you're saying you said something about plasticity taking about 20 to 30 years. Does that mean for it to start degenerating again? What was that timeline?
No, I meant 20 to 30 years to replace all tissues of the brain.
I see.
Because we do little areas, scattered little areas at once, maybe we do, 10, 15% of the brain at once that we slowly silence in the presence of new tissue. And we have to do this a few times to finally have replacement of all the tissue in the brain. So that's the 20 to 30 years. But at that point, you will have the brain of a 20 to 30-year-old. And so you'll be okay for another 50 years if you want.
Mm-hmm. So then what do you think would be a good age to start this progressive replacement program?
I hope to start it when I'm 70.
Mm-hmm.
Ideally with replacing other body parts as well,
right.
Because especially recovery from surgery as we get older gets harder and harder.
Yeah.
But if at the same time we're becoming younger and younger, then we can recover better and better from the surgery. So yeah, I to be honest with you, I could probably use more tissue right now because I feel I'm like forgetting things, or but I can wait a little too.
Plasticity, that's the answer.
Yeah.
More efficiency, Hebert. So yeah. What are some of the biggest challenges in this that you see?
I guess there's challenges at different levels. I mean, there's the engineering challenge of making this tissue functional.
Mm-hmm.
The surgery as well is not something that's going to be easy. Surgery never is. I mean, brain surgeons do brain surgeries all the time. They're super good at it. They remove tissue that's epileptic, for example. So again, to them, this is not a big deal, but if you're going to need to do like 6 rounds of this surgery, you want to be sure that whoever is doing it is very skilled. And so I think at some point, having smart robotic, surgeons, but also that, can sort of bioprint the tissue in place, which is what we're doing now, except we don't use a bioprinter. My PhD student does it by hand, sort of layers the cells in to get the right cytoarchitecture, the right layering of the neurons. But that might be something that needs to be more streamlined to be applied more broadly in patients and humans. So first, engineering the tissue, I think, will have its challenges, but also the surgical implementation.
Mm-hmm.
And then, you know, I think for the brain, it's a pretty, I think we can get, you know, I don't think there'll be political roadblocks so much, because we're gonna, you know, our first clinical indication would be something like stroke. And it, you know, when you have a stroke, you lose the functions that are there because there's no time for plasticity to move them to another part of the brain. Right? So that's a stroke is a catastrophic event. So it's a little different than aging in that sense. But our tissue that we put in is immature. So it's the equivalent of a 1-year-old tissue. And so that should allow, because, you know, 1-year-olds, 2-year-olds can learn language without any effort. So there, it's like plasticity on steroids. It's like this blank slate just waiting to take on function.
Mm-hmm.
And so putting that in the brain of a stroke patient where they've lost tissue due to stroke, I think will give them, you know, this wonderful ability to relearn lost functions, whether that's language or mobility in the contralateral side of their body. So that will be our first clinical indication. And there's really nothing that can be done for these patients as is. So as long as we can demonstrate safety, which other groups are doing for other reasons with brain transplants, I think we can implement that in the clinic. And then if we see benefits from that, then there are other forms of sort of accelerated degeneration or dementia that are also untreatable. And you know, I think strong arguments can be made for why patients would need this tissue. And so I think we can progressively move from, you know, something like stroke to forms of dementia to aging, because, you know, the difference between dementia and aging, it becomes a fine line, right? And so I think the, you know, the roadmap to getting there for the brain is pretty clear. You know, then I think what the whole longevity field faces in terms of complete rejuvenation for the whole body and the brain, there might be more of a challenge there, you know, to get this sort of generally accepted. Although I think all it will take is, you know, somebody to do it on their own, and then everybody's going to want to do it, right, if it works. So I don't know. I don't know how challenging that'll be, but that could be a challenge to get it to that point where we can actually do it, and it is sort of accepted as something we all do. Yeah.
Because at that point, I can see there being, you know, some pushback or controversy around, you know, you're still functional and healthy. Why do you need this? But then I think, as you said, if we do start with people who have brain disorders or stroke or dementia, I think it could be a very solid pathway to get there. That if it works for them, and you start seeing your brain just declining in function with age, why not prevent it early? So
yeah,
I'm sure there's promise. Yeah.
And if I could add just one thing, maybe too, is that, you know, I think I believe fundamentally that everybody should have equal access to the best healthcare that's out there, as well as the best education that's out there. And clearly we don't have that now, and that's a problem already. So, you know, there's a lot of inequity in access to healthcare as is today. And a lot of us, you know, vote against that, or and some are much more active in fighting that, but it's you know, it's very difficult. I worry that, and I think we should consider how to go about, you know, getting around this problem for age reversal as well, because I think that, if anything, could, you know, exaggerate the differences between those who have access to these treatments and those who don't. And so I think we should really take steps early on to minimize that inequity and to try to give access to these rejuvenation therapies to everyone. And what I'm talking about, these types of replacements, of course, a lot more expensive than taking drugs, but they also save money because if you're really avoiding the chronic diseases of old age, which are hugely expensive, you can easily pay for these replacement therapies. So it just will take the social and political will to get that to that point.
Yeah.
But hopefully we do.
Yeah. I think that's a great point, but I agree with you. I mean, these problems will come up. Hopefully, we're going to solve them in an optimistic and rational way. At least I'm optimistic about it.
Yeah.
I think one other question on just your research I had was, so you're saying in theory, if I do this full body replacement and also progressive replacement of the brain, I could have the brain of a 20 or 30-year-old when I'm much older and also retain my memory and other functions that I've learned in the previous 80 years, 90 years, whatever it is. So that means, I mean, that could be really exciting because then I can essentially, I have learned from my past previous 80 years, I have all this experience, but I can also go out there and learn tons of other new things, start different careers. Basically, there's nothing stopping us at that point from learning as much as we can because we should have that capacity again, right?
Yes, well, we will always have the capacity to learn new things. That's the nature of the brain. There's a trade-off, though. I mean, this plasticity, if if there are things, functions you never use, you do forget them, and that that's how it works, right? And that's happening to all of us now. We're forgetting things not just because we're getting old, but because we're not using them. And so, yes, you can learn new things to a certain extent because at some point you'll start, forgetting other things as well. So, but you'll have the choice. You want to learn new things, you can learn new things.
You'll have a more efficient brain then.
Well, the younger one, yeah, you'll be able to learn much more than the older brain. Yeah.
Yeah. Oh, that's great. I think two last questions I want to leave you with. I mean, why do you think there aren't other people working on this approach? Because I mean, I'm sure there are more people I'm not too familiar with, but then it just seems like a lot more people are working on developing drugs as opposed to the approach you're taking.
Yeah, well, there's a lot of motivations for drugs. I mean, the pipelines are there, the profits, you stand to profit a lot more, a lot quicker from that. You can get, they're more readily, you can get insured much more easily for that. So companies have more incentive to develop them because they'll get their money back. So there's a lot of reasons why drugs are attractive. But in terms of tissue replacements, I think we're slowly moving towards that for diseases in general, because you can provide a cure as opposed to having to take medication forever and not really solving the problem. So tissue or organ replacements have the potential to provide a cure. And there are many labs working on these, developing organs, lab-grown organs and tissues for the purpose of treating either damage or disease. And so progress is being made. There are far fewer for the brain. Again, there are many labs doing transplants of cells in the brain, but in a way to get a functional tissue, there aren't very many at all. I think we're one of the only labs, and I wish there were hundreds of other labs. But the reason is it's a big project, and it's not easily funded by typical funding agencies like the NIH in the U.S. or other typical funding agencies in other countries. Because it's such a big project, because it's such long term, we're collaborating with over a dozen other labs to get the right cell types to be able to build this tissue. So it's a very big project with a lot of moving parts. And so I think most scientists in my area of brain regeneration are not in a position to undertake such a big project.
Right. Yeah, that makes sense. I mean, it is a bold project, but you've at least convinced me that it's an extremely important one, and one that could actually really cure aging and beat it. So I hope others have been convinced too.
Yeah, there are some out there.
Right. And I think progressively, hopefully more so in that direction. So I guess for anyone who will be watching this or listening to this, is there something that you're seeking help on that potentially someone could reach out and help you?
Yeah, well, we're looking for highly specialized personnel, and we will be more and more. So, somebody with tissue engineering experience, working with human stem cell, especially the patient-derived, induced pluripotent stem cells. So we have protocols for making all the brain cell types that we want, but they would need to be optimized for clinical, to be better used in the clinic. So we need computational biologists, we need systems neuroscientists that are capable of analyzing large data sets of neuronal activity and making sense of how different brain parts are communicating with each other. We need electrophysiologists. And we need, people working with different animal models, not just rodents, but also, humans, primates. So it's a huge endeavor.
Yep. All right. So great. Whoever's been convinced by your theory, go work with Professor Eber. It's going to certainly help us cure aging. Lastly, I guess, is there anything else that you want to touch upon that maybe we haven't covered?
I think we covered pretty much everything. So thank you very much for having me. You had very good questions. I like the order of the questions because it really helped me build the case. So I appreciate that. You made it easy for me. Yeah, thank you very much.
I really enjoy this conversation. As I said, I've definitely been more convinced by your approach. So thank you for doing all the work you're doing, and thanks for your time today.
Yeah, thank you.
Hi everyone, if you enjoyed today's episode and learned from it, I would love it if you could subscribe to the channel and like the video. It helps get the word out. Additionally, it would mean a lot if you could show some support and share the channel with your friends and family. I want to get the word out on aging and longevity science to as many people as possible, such that we can all be healthier for longer. Okay, if you enjoyed this content on longevity science and science-backed health optimization and longevity lifestyle hacks, you can check out my newsletter, which is livelongerworld.substack.com. You can also listen to the audio version of this podcast on Spotify or other places that you get your podcast if that's the preferred medium. You will find all these resources on my website, which is livelongerworld.com. All right, I do have social media as well. So if you want to follow me on Twitter, I am at livelongerworld. Instagram, I am at Longevity Future, where I often post graphics distilling the basics of longevity science and also talking about longevity lifestyle hacks that I personally practice. Lastly, you can support my work by donating on Patreon, which is simply patreon.com/livelongerworld. All of this is also included in the show notes, so if you want, check that out. And that's it for now. Stay in good health, and I will catch you next time.